There was a time, not too long ago, when we associated engineers exclusively with the design and building of inanimate objects. Booster rockets and jet aircraft, bridges and skyscrapers, automobiles and heavy equipment, computers and other technologies like radio telescopes, and medical imaging equipment like MRIs and PET scanners. But now, engineering has moved beyond the inanimate world into the world of animate living organisms. Engineers working to create inanimate objects develop blueprints that are then used by other workers to build the object, be it a computer, an aircraft, or a skyscraper. In their laboratories, the engineers of living organisms, or genetic engineers, manipulate what is often referred to by biologists as the blueprints of life, an organism's genetic material, its DNA. Once this is successfully accomplished, the organism, unlike an inanimate object, actually builds itself cell by cell, following the new blueprint exactly. No construction or assembly line workers are required to complete the projects of genetic engineers. But genetic engineers can't effectively redesign and modify living organisms without first reading and understanding their original blueprints. Thus, for some time, geneticists have been trying to map the DNA of various organisms important to biological research, including humans. Mapping the DNA or genome of humans consists of identifying which genes along our 23 pairs of chromosomes control which human characteristics and the sequence of nucleotides within these genes. The early focus of such research was on locating and sequencing the genes responsible for serious genetic disorders such as cystic fibrosis, sickle cell anemia, and Huntington's disease. This information has helped physicians develop genetic tests to identify carriers and those afflicted even before birth, and may eventually allow geneticists to replace defective genes with properly functioning ones through a technique called gene therapy. In 1990, the ambitious goal of mapping the entire human genome was officially begun by scientists and laboratories throughout the world. The work of these scientists and laboratories was collectively known as the Human Genome Project. After 13 years and intense competition between public and private interests, mapping of the human genome was largely completed in 2003, providing a solid foundation for genetic research for the rest of the century. In addition to mapping the human genome, geneticists have also mapped the genome of a variety of organisms important to biological research, such as strains of E. coli and yeast, as well as species of fruit flies, mice, zebrafish, and various plants. One of the interesting findings of this mapping of genomes is that there isn't necessarily a correlation between the amount of DNA or the number of genes an organism has and the complexity of that organism. For example, there are flowering plants that have 40 times more DNA in the nuclei of their cells than do humans. And while rice plants have only 1/6 of the DNA of humans, they have over twice as many genes, 60,000 as opposed to the roughly 25,000 found in humans. That 25,000 genes, in turn, is far fewer than the 50 to 100,000 genes geneticists thought humans had as recently as a decade or two ago. Armed with information they have about the genes and genomes of various species, genetic engineers have begun taking genes from one species and splicing them into the genome of another. This is referred to as recombinant DNA technology. Recombinant DNA technology has a number of uses. One is modifying an organism so that it produces a material, such as a protein, that is useful to humans. For example, some humans don't produce enough of the hormone insulin that helps cells of the body metabolize sugar. This leads to the disease diabetes mellitus that is eventually fatal if left untreated. Treatment of this type of diabetes takes the form of injecting insulin up to 4 times a day. Until recently, the source of insulin had been the pancreases of slaughtered pigs and cattle. However, recombinant DNA technology has allowed genetic engineers to place the human gene that codes for the production of insulin into bacteria, causing the bacteria to produce human insulin. Likewise, genetic engineers have developed sheep that produce the human protein AAT in their milk. AAT is often used in treating the relatively few individuals who develop emphysema as a result of a genetic defect that leaves their bodies unable to produce enough AAT. AAT protects the lungs from the enzymes used by white blood cells to defend the lungs from invasion by outside microorganisms. Without the protection of AAT, the enzymes released by white blood cells cause the same breakdown in the tiny air sacs or alveoli of the lungs as does cigarette smoke or exposure to other airborne irritants. Another use of recombinant DNA technology is to genetically modify the characteristics of organisms in a manner that either increases their commercial value or simulates the characteristics of human diseases in experimental organisms so that researchers can study various treatments in more controlled conditions that would be possible with human subjects. One of the most obvious areas in which to increase the commercial value of organisms is in agriculture. For hundreds of years, farmers and ranchers have been trying to increase the yields of crops and farm animals through selective breeding. In other words, controlling the passing of genes and their attendant characteristics from one generation to the next by controlling which organisms breed with one another. But now, genetic engineering has taken that a step further by allowing the desirable characteristics of one species to be transferred to another. For example, the bacterial species Bacillus thuringiensis produces a protein that is toxic to insects. The gene for this protein has been inserted into certain species of corn plants by researchers, and initial results show a large reduction in the damage done to these strains of corn by a major corn pest, the European corn borer. Similar efforts have been made to create pest-resistant strains of cotton and rice. Perhaps even more fascinating is the insertion of a gene found in a particular species of flounder into potatoes. The gene taken from the flounder produces a protein antifreeze that allows the flounder to operate at sub-freezing temperatures. Potatoes are often grown in rather cold environments, such as high mountain valleys in both North and South America. Initial research by the International Potato Center has indicated that the flounder gene significantly increases the frost resistance of potato plants. Organisms, such as the corn and potato plants we just looked at, are referred to as transgenic, as they are made up of genes from at least 2 different species of organisms. Genetic engineers have also produced transgenic farm animals. Danish researchers are trying to engineer cows that are resistant to udder inflammation by inserting a human gene that produces a protein found in human breast milk that helps infants fight infection. It is hoped that the protein will help prevent infections and inflammations of the udder in transgenic cows. Australian researchers are working to introduce a gene found in a number of plants into sheep. The gene codes for an enzyme that destroys insects by breaking down the chemical chitin that is a major component of their exoskeletons. Researchers hope that this will produce sheep with an insecticide of sorts in their skin, making them immune to lice, blowflies, and other insect parasites, and thus saving sheep ranchers millions of dollars by avoiding the costs associated with dipping sheep in insecticides. Transgenic rats and mice have been engineered that contain the genes for various human genetic disorders, such as cystic fibrosis, muscular dystrophy, sickle cell anemia, cancers, and others, so that these diseases can be studied in the laboratory. The first of these were the so-called Harvard oncomouse that were developed at the Harvard Medical School and patented in 1988. These strains of mice were developed specifically to be used in cancer research, and millions of dollars were spent on their development. The millions of dollars were to be recouped by charging royalties to pharmaceutical companies that developed anti-cancer drugs using the mice. To date, however, for whatever reasons, no pharmaceutical companies have signed up for deals using the mice. More successful are the SCID and SCID-HU mice developed by Systemic. SCID mice have their entire immune system knocked out and are used to study the human disease of severe combined immunodeficiency, in which infants are born without immune systems and are often referred to as bubble babies by the lay public, as they are often protected from invasive microorganisms by living in a bubble-like device. SCID-HU mice have a human rather than a mouse immunological system. Thus, they can be used to study the response of the human immune system to a variety of illnesses, including AIDS. Before going any further and looking at the accomplishments of genetic engineering, we should perhaps now look at the tools genetic engineers use to change the characteristics of organisms. For example, how do genetic engineers create bacteria that produce the human hormone insulin, or create mice with human immunological systems? In order to alter an organism through recombinant DNA technology, genetic engineers must be able to insert a new gene or genes into an organism's genome. They do this by first breaking apart a strand of the organism's DNA, and then inserting a new gene or genes from another organism into the break in the DNA. The cutting apart of genetic material or DNA is accomplished with the use of what are called restriction enzymes. Many bacteria produce restriction enzymes in order to cut up the DNA of viruses that attack the bacteria. Restriction enzymes recognize and cut DNA at particular sequences. For example, the restriction enzymes scientists call EcoRI always cut DNA at the nucleotide sequence TTAA, breaking the bonds between adenine and thymine nucleotides. Genetic engineers have identified hundreds of different restriction enzymes. Each restriction enzyme is very specific and will only cut the section of DNA that it recognizes. You will notice that in the strands of DNA cut by a restriction enzyme, there are so-called sticky ends left. In the case of EcoR1, those of the TTAA end are complementary to those of the AATT end. If the correct repair enzymes are present, the two pieces can be rejoined. However, it would serve no purpose to rejoin two pieces of DNA just cut apart. Rather, genetic engineers take one of the sections of DNA just cut apart by the restriction enzymes and insert them in another chromosome or similar structure that has been cut apart by the same restriction enzyme and therefore has the same TTAA and AATT sticky ends. Having the same complementary sticky ends, the two pieces of DNA join to form a new genetically different strand of DNA. Bacteria have been helpful to genetic engineers not only in providing them with restriction enzymes, but also with what are called plasmids. In addition to their single chromosome, many bacteria also contain tiny rings of DNA called plasmids. Plasmids are usually about 1,000 to 100,000 nucleotides long and act as independent, self-replicating molecular operators within bacteria. Plasmids are one of the ways that DNA recombination goes on within nature. While a bacteria's chromosome contains all the necessary genes to code for the bacteria's existence, plasmids provide an effective way by which traits not contained in the chromosome can be passed from bacteria to bacteria. For example, some plasmids contain genes that code for enzymes that digest certain antibiotics, such as penicillin or ampicillin. This is obviously an advantage to the bacteria. When a bacteria containing these plasmids dies, it breaks open and liberates these plasmids to the outside environment, and they are often taken up by other bacteria that then acquire the traits coded for by the plasmids. Genetic engineers can place liberated plasmids in a solution with restriction enzymes and cut them apart. The engineers then place the gene segment they wish to insert into the solution with the plasmids, and then add repair enzymes that join the pieces of DNA together at their sticky ends. These new plasmids, with their newly inserted genes, are then placed with bacteria into a different solution that enables the plasmids to readily penetrate through the cell wall and membrane of bacteria. The bacteria that are exposed to the plasmids are taken out and grown in a culture for a short time. If the plasmids into which the experimental gene, such as the gene for the production of insulin or growth hormone, is inserted, also have genes that provide resistance to ampicillin and amoxicillin, a unique technique can be used. Genetic engineers know that not all the bacteria exposed to the plasmids will absorb them. By exposing the cultured bacteria to antibiotics such as ampicillin and amoxicillin, they kill off the bacteria that didn't absorb the new plasmids, while those bacteria that did absorb the new plasmids containing the genes for antibiotic resistance and for, say, insulin or growth hormone continue to grow and thrive in the culture. Scientists continue to grow these bacteria until the insulin or growth hormone produced by them is sufficient to be extracted and purified for use by human patients. But how are genes inserted into the chromosomes and DNA of more complex organisms such as plants or animals? There are two major methods of placing DNA into the nuclei of eukaryotic cells: vectorless and vector methods. Vectorless methods of insertion of DNA don't require the use of a third intermediate organism to deliver the DNA from one species to another. Vectors, in the biological sense of the word, are organisms that carry the DNA of one species and insert it into another. There are a number of vectorless means of inserting DNA into cells. One commonly used method of inserting DNA into cells is firing tiny metal particles mixed with the DNA to be transferred into recipient cells. This method is referred to as biolistic delivery. However, there are a number of problems with biolistic delivery. One is that damage can be caused as the result of the metal particles fired into the cell. Another, more intractable problem is that only a small proportion of the fired-upon cells take up the foreign DNA. A second method of vectorless transfer is microinjection, in which foreign DNA is injected directly into the nucleus of a cell. This approach is often used in the genetic engineering of animals. Often, hundreds of copies of a given gene are injected into the fertilized embryo of an animal in the hopes that some of these genes will be taken up in the genetic material of the developing embryo. A third method of vectorless transmission is called electroporation. In this process, the cells to be genetically engineered are placed in a solution of foreign DNA and a strong electrical field is applied. The electric field creates pores in the cellular membranes of cells and leads to them taking up the DNA in the solution. Electroporation is frequently used in the genetic engineering of plant and fungal cells. Recently, an even simpler way of getting foreign DNA into plant cells was discovered. Water, plant cells, foreign DNA, and a chemical called silicon carbide are placed together in a test tube. The test tube is shaken, and the crystals of silicon carbide punch very small holes in the plant cells through which the foreign DNA can enter. A high percentage of the new DNA introduced into plant cells in this way is eventually inserted into the plant's existing DNA. A vector is any agent that acts like a carrier or transmitter. The three major vectors used by genetic engineers are bacteria, viruses, and liposomes. One vector that has proven effective with a number of plants is the bacterium Agrobacterium. Agrobacterium is a soil bacterium that naturally attacks certain plants and causes a disease known as crown gall disease in the process. Agrobacterium infects these plants and causes the development of swellings or growths referred to as galls. Scientists found that these growths were caused by the bacterium inserting part of one of their plasmids into the DNA of the host. Scientists labeled this plasmid as TI, for tumor induction plasmid. Subsequent research found that a mutant form of the TI plasmid exists that does not cause crown gall disease, but still inserts its DNA into host plants. This ability of Agrobacterium to insert new DNA into the chromosomes of host plants is not wasted on genetic engineers who have inserted new genes into the plasmids of Agrobacterium, which in turn have inserted the new genes into the DNA of plant cells infected with Agrobacterium. Certain viruses, such as retroviruses, can do something very similar to Agrobacterium. They synthesize complementary DNA from their RNA and then insert this into the host DNA. Retroviruses have been used in genetic engineering and in research on humans. For example, retroviruses have been used to insert a functional copy of a faulty gene into bone marrow cells. Bone marrow cells give rise to our blood cells and can be responsible for a number of genetic disorders, such as sickle cell anemia. While retroviruses are good candidates for this therapeutic approach, as they have millions of years of experience in inserting their genetic material into host DNA, there are a number of drawbacks to using retroviruses. One of the problems is that retroviruses only affect dividing cells. So there is a limit to the number of diseases for which retroviruses can be used as a vector. For example, many human diseases, such as those of the nervous system, are not caused by mutations of dividing cells. Another difficulty with using retroviruses is that they insert their DNA, along with the gene to be inserted, more or less randomly in the DNA of the host. This has two major consequences. The first is that the newly inserted gene may not work as effectively as it otherwise would because it is not near other genes that would normally help turn the inserted gene on and off. A second and more dangerous consequence is the accidental insertion of the new gene into what are known as tumor suppressor genes. As their name suggests, these genes help prevent cancers. Disruption of the tumor suppressive gene by the insertion of a gene through the activity of a retrovirus has been shown to sometimes lead to the development of cancers in test monkeys. For these reasons, researchers are also looking at the use of other viruses, such as adenoviruses, to insert functional copies of genes into host cells. Adenoviruses are unlike retroviruses because they do not integrate their genes into the host's DNA. This results in advantages and disadvantages. The advantage is that there is no risk of the adenovirus inserting its genetic material into the host cell's DNA in such a way as to disrupt its normal functioning or even causing cancer. On the other hand, the disadvantages are that these genetically engineered cells do not pass a new functional gene along to their daughter cells, as is the case with genes added to the DNA of retroviruses. This means that once the genetically engineered cells die, the production of the corrected protein or enzyme ceases. Thus, engineering cells in an individual via this process would necessitate repeated treatment based on the life cycle of the cells. Another approach to introducing genes into cells is the use of hollow membranous spheres called liposomes. Liposomes are made out of the same phospholipid materials that cell membranes are made out of. Thus, liposomes are able to easily fuse with the membranes of cells and release their contents into their cytoplasms. The disadvantage, however, is that most of the genes introduced into cells in this way do not end up inserting themselves into the cell's DNA. liposomes have been used in experiments with individuals with cystic fibrosis. The liposomes containing the correct versions of the genes responsible for cystic fibrosis are sprayed into the respiratory system of the individual and then fuse with the cell's membranes and release their DNA into the cells. Early results have been encouraging. Scientists have found that only 10% of the cells lining the lungs of a cystic fibrosis patient need to have the correct genes and produce the otherwise missing proteins in order for the lungs to function normally. As has become obvious from our previous discussion, genetic engineers often need to be able to produce large quantities of the genes they wish to insert into a given individual. Genetic engineers make these copies through a process referred to as polymerase chain reaction, or PCR for short. The first PCR machines were an incredible boon for genetic engineers. Polymerase chain reaction machines create copies of genes in a number of steps. First, a strand of DNA containing the stretch of DNA to be replicated is heated to about 98 degrees centigrade, or just below the boiling point of water, which causes the DNA to separate into two single strands. Primers in the mixture bind to complementary regions of DNA where the engineers want the replication of the DNA to start. An enzyme called DNA polymerase takes free nucleotides in the solution and uses them to replicate each DNA strand from the primer to the end of the strand. The result is one complete copy of the gene to be duplicated, plus any nucleotides extending from the gene to the end of a given strand. Again, the newly formed DNA molecules, like the original strand of DNA, are heated to 98 degrees centigrade and separated into single strands. This time, however, both the original section of DNA and the first cycled copies of the DNA are used as templates. When the first cycled copies are used as templates, the newly synthesized DNA strands start at one end of the gene and finish at the other end. No extraneous DNA is copied. The third cycle begins just like the first two. Note that in this cycle, 2 of the 8 strands to be duplicated include only the nucleotides of the gene scientists want to copy. Therefore, this third cycle of PCR synthesizes 2 strands of DNA that are exact copies of the gene, with no superfluous DNA at either end. Further cycles of the PCR continue, and the proportion of DNA copies that consist of only the gene nucleotides rapidly increase. In the fourth cycle, 8 out of 16 complete strands of DNA include only the gene of interest. In the fifth cycle, 22 out of 32, and so on. As each cycle takes only a few minutes to complete, within a few hours, PCR can produce many billions of copies of a given gene, most of which have only the sequence of nucleotides specific to that given gene. The vast numbers of a particular gene created by PCR can then be inserted into plasmids and other vectors. This ability to reproduce huge numbers of genes and transfer them between species has provided genetic engineers with an unprecedented power to change the face of life on Earth. A power that, as we have seen, promises many positive benefits for human society, but which also threatens to confront us with difficult environmental, economic, moral, and ethical issues that we as a society can only deal with effectively if we have a thorough understanding of the science that underlies the engineering of life.